Okay, I just want to welcome you all to our final session of the Arthropod Genomics Symposium. This one will be on agricultural genomics. I want to say a special thank you to all of you for attending. This has been a wonderful AGS this year with so much great support and so much great participation from all across the world. Just a reminder to everyone, there's 3 ways that we'll be dealing with questions. What we would like you to do is post your questions within the Q&A feature that you'll find at the bottom of your Slack, or I'm sorry, at the bottom of your Zoom session. Please use that instead of the chat function. And when you're doing that, we've posted some instructions within the chat to please address your questions to the speaker. So if there's time at the end of the speaker's talk, we'll answer some questions then. For questions that we don't get to then, we'll try to address those during the Q&A at the very end of the session. And if we don't have time then to address all of the questions will be posted onto Slack, and there'll be space there for you to ask and answer questions. There also, at the end of today, will be a separate Zoom session where we can have some additional conversation. So with that, I want to hand it over to Surya to get us started on any other housekeeping points.
Great. I think Anna covered everything already. Most of you know about the questions. Please just upvote questions and don't repeat them. If a similar question has already been asked, the chairs will, as in we, will deal with the questions at the end of the talk if there is time, or we will have a Q&A session at the end. And then there is the all speakers chat on Slack, where all these questions will be posted. So you will have many chances of interacting with the speakers, and this Slack will be available after the meeting. We will talk about it in the closing remarks. And back to Anna to introduce the first speaker.
Yep, so with that, let's get started with our first speaker, Scott, who's joining us very early this morning in Hawaii to speak about the Ag 100 Pest Initiative through USDA.
The host disabled screen sharing still?
Oh, I didn't fix that, sorry.
That's okay.
Yeah, okay, that should work.
Yeah, thank you for having me come and represent the Ag 100 Pest Group. Given the limited amount of time today, I'll be spending most of the time on sort of what strategies we're using for sequencing, the diversity of different insect pests that we're doing. Hopefully some of these methods and techniques, you can utilize in your own labs and try out as well. So what is the Ag 100 Pest Initiative? It's a goal within USDA ARS to produce annotated high-quality reference genomes for the top US arthropod and agricultural pests. It also includes some beneficials. It's USDA's commitment and contribution to the Earth Biogenome Project, and you can go to this link here and see the species that are nominated at this point for the project. It's not just me, not just Anna, but a large group of people across the USDA. This includes a leadership group, and then different people that work at different steps of the process, things like tech transfer, getting the samples, extraction, sequencing, assembly, and post-assembly and curation. Selection from the top pests hitting at least 10 orders. Nominations are gathered across different agencies, APHIS, things like ITAP and CAP surveys, USDA researchers, so what do researchers within USDA need? And then also the community here. Represents diverse nominations, including pests of crops, animals, bees, trees, storage pests, foreign pests that aren't here yet, and there is no one fit. So it could be a giant beetle, it could be a tiny mite, it could be anything. So really presents some unique challenges to the project. And so really this is a community effort. So, our goal is basically to sequence every pest insect species that's nominated. And ideally, we'd like to target chromosome scale diploid assemblies for these pests. This requires multiple library types. And what we're using today is a config assembly that's supported by PacBio hi-fi data And then scaffolding information, usually coming from high-C data. Sometimes this could be coming from bionano or linkage maps as well. We're following the Earth Biogenome Project quality standards, shooting for a 3.4.2 QV40 standard. So that represents a 1 megabase contig N50 and a 10 megabase scaffold N50. EBP is moving sort of beyond this, but this is kind of where we started the project and initially what we were trying to hit. And then we're trying to hit everything to some level of chromosome assignment, usually coming from high-C data, and a consensus accuracy of QV40. So some of our limitations are obviously the small physical size and limited DNA material that's going to work for these various methods, and then the extremely wide diversity of taxa, tissue types, sizes, etc. So a large portion of our samples fall below the typical standard library prep for things like PacBio. So our real standard is really more of a low input sample. And throughout the project, we kind of transitioned technologies and approaches. So when the project started, the state-of-the-art was using PacBio Sequel 1 instrument. That's the 1M cells, CLR long reads. And these are reads that will need Illumina polishing, things like the Falcon and Canoe assemblers. These take days or weeks of wall time per genome on a compute cluster to generate an assembly. And what you need for that is really good DNA. Bigger DNA, the better. Trying to get long reads. Those long reads will represent more output. You also need a lot of DNA, or at least relative today. So we're talking microgram amounts. One way that we got around this was to do unsheared DNA library preps with no size selection, just trying to capture the longest reads possible. Today, we've sort of migrated. The state-of-the-art is the PacBio Sequel 2 instrument with 8 M cells using hi-fi circular consensus reads, no polishing needed, and hi-fi specific assemblers. And these can produce genome assemblies with under 1 day of compute time. So the need for these, we're using modified low input methods using nanogram amounts of DNA. So our typical standard low input would be 200 to 300 nanograms or more of DNA. And we shear the DNA to 10 to 20 kb. So the output is not dictated as much by the length of the DNA going in. Also, this allows us to use samples with smaller DNA input sizes. It still has to be good DNA, but you don't benefit from extracting 100 kb DNA maybe over something like 50 kb. So our standard library prep workflow right now is our samples are typically sent to us snap frozen as fresh material. We sometimes partition some of the sample to make a high-C library. So if it's something the size of a housefly, we would maybe take the thoracic tissue and make the PacBio library just from the thorax, and then use the rest of the sample to make a high-C library. We grind the sample to a powder using the GenoGrinder and a steel bead. We use DNA extraction by hand using the QIAGEN MAGATRACT high molecular weight kit. And then we do a lot of DNA QC before going into library prep. So doing things like sizing on fragment analyzer where you don't want to see any shearing of the DNA. We figure out how much of the DNA it is using a qubit or Genomics sort of probe-based methods. And then we look at purity pretty carefully, doing the 260-280 ratios, looking at 230 for peaks of contaminants and things like that. And basically, if the sample's too smeary or degraded, stop, don't continue, start over, do something else. And if it's too dirty, we have some methods for cleaning up the sample further. And if you're interested in that, you can reach out to me. So that extracted DNA is sheared to hi-fi library size. We've been using Diagenode Megaruptor equipment to do that. It seems to be pretty good at generating very tight fragmentation profiles. So when we're not size selecting the library, we're not getting a lot of tails of smaller DNA or larger DNA. And then making our libraries with the SmartBell Express 2.0 kit. And they have this extra enzyme cleanup step that you can do. It's an extra reagent you buy. It basically destroys any degraded or imperfect libraries. And I highly recommend using that step so that you know that the library you're making and putting in, all of it is going to start making sequence as well. So this is just a hi-fi approach. I'm not going to go through it in too much detail, but basically you're taking an amplicon and sequencing around the circle over and over again to get this highly accurate read. So if our samples are low input, we do no size selection. And if it's a standard input, we could size select on something like a Sage Pippin, Blue Pippin, or a Sage Elf. And we load and run these on a SQL2 system, 70 to 100 picomolar loading, 30-hour runtime. So what does this look like in practice? This is an example of a low input sample. You can see the input DNA here with the trace, and you see very little shouldering coming off of the peak. Really doesn't matter what this peak size is. You just don't want to see any evidence of degradation. So different kits might give a different size here. The critical component is that there's no degradation. So shearing on the Megaruptor, you can see a very clear shear. This is at 17.5 kb. A little bit of shouldering, but that only represents a small amount of this sample. And then when we make the final library, we can see that it emulates the shear profile of the post-shear DNA very closely. And during the protocol, there are some steps to remove DNA smaller than 3,000 base pairs. If you're seeing a hump here, again, I wouldn't continue with this. It's going to give you a lot of small fragments away from that target insert size that you're generating. So by not doing size selection, we don't get any sample loss, which is our way of being able to go in with low inputs amounts of DNA and still get good runs. So this is an example of a hi-fi run performance. One run here generated 164 gigabases of data. This one generated 260. And then when you go around the circle, right, you're only generating a unique molecule for that. And the coverage of that is, this is 30 gigabases or 50 gigabases of data. And you can see the mean here, this top one was that one I showed in the previous, is very close to the length of the library going in. So we have an N50 of 18.7 kb. So one run is sufficient for a genome of 500 megabases or so, or potentially larger. And a library yield may allow for multiple cells from the same prep. So you can do several cells if your genome size is bigger. So the species we've done today, and I'm not going to list them here, but you can go to this link and see the list of the species nominated. We're working to get the config data as well uploaded to that link. And this is our original CLR. Oh, I gave it away. This is our original CLR data here. So this is using the SQL1 or SQL2 Falcon assemblies, purge haplotics to get a haploid assembly, correcting the data with PacBio and Illumina, and a lot of compute time. And you can see from genomes as small as half or a quarter of a gigabase up to 2.5 gigabase. Most of our assemblies are running in this 1 to 3 megabase N50. We seem to be on a ceiling for some reason at not being able to achieve anything higher than 9 or 10. But we thought these were very good. They were in very few pieces relative to the N50 size and have shown to scaffold pretty well as well. When we moved to hi-fi data, we initially only piloted samples that were 500 megabase or smaller. So that's why most of our dots are over here. But what you can see here is we're running these all in a single SQL2 run or half of a run, hi-fi ASM for assembly. You're reaching Q40 straight out of the assembly. It's generating a haploid or phased assembly as well. And the compute time is under 1 day. And you can see here we are generating N50s up to 25 megabase. And a lot of these are capturing near chromosome size assemblies. So a big difference between the CLR approach and the hi-fi approach, at least in what we have seen. So some examples of this I'm just going to go through very quickly. The Indian meal moth is thought to have 30 autosomes and a sex chromosome. We generated a library. This was one of our earlier ones. Our shearing was smaller. We were still getting comfortable with the approach. We have 52 gigabases of unique yield on this. And running it through hi-fi ASM, the assembler, it assembled into only 88 contigs. And if you look at the N90 here, there's 35 pieces representing 90% of the genome. And it's thought to only have 30 autosomes. So that's nearly 1 contig for every chromosome, a little bit more than that in reality. 99.7% of the genome is in 50 kb pieces or larger. So we can validate that. We're generating high-C data for our assemblies using this Arima Genomics kit. And you can see here very strong high-C data. And when you don't see any yellow here, that means there's 1 contig encompassing the entire chromosome. So essentially,
2 more minutes.
okay, I'm going to move. Next one is the Diarrhea beetles. These are weed-feeding biocontrol beetles, testing 4 species. They're all 425 megabases in size. N50s here, again, we're getting 22, 23 megabase N50s, only in a handful of pieces. So worked out really well. We kind of moved from that into some smaller insects with low DNA yield, below 200 nanograms, but they have relatively small genome sizes. So what we're doing here now is barcoding 2 libraries on a single cell. This allows for not only being able to run small things, but also being more cost efficient as well. So by combining 2 libraries together, you reach the molar loading concentration you need. And these assemblies came up really well. This tribolium beetle, for example, is 66 pieces, 90% of the genome, and 50 of 14 megabase. So we then kind of went into things bigger than 500 megabase, shooting for 2 flies. Shenna Sim presented on this on the first day. There are these 2 guys. We don't see, you know, as you go bigger, the data is getting worse. We did run 2 cells on this to get optimal coverage. But you can see here that there's only a handful of contigs spanning each chromosome in these assemblies. So these N50s are 50 megabases and 70 megabases or higher. We also have many samples below the standard input. Many are beneficial insects we want to do. So these are less than 100 nanogram DNA yield, beetles, thrips, all sorts of little things. PacBio, we're beta testing an amplification-based kit with them. So this goes inputs with 5 to 20 nanograms of data or DNA. Trying it out with a sawtooth crane beetle. It's not that small of a beetle, but it's very flat, very low DNA yields. So we tried a standard input and it was pretty poor, N50 below 100 kb. The ultra low library prep starting with 5 nanograms of material, amplifying that DNA and then making a library from that. We get an N50 of 1.87 megabases. And more importantly, the BOOSCO coverage is much better. So here we were losing genes. Here we basically have a representation of all the genes in the genome. We're doing some other small insects now. This is still definitely a work in progress. Seems to work well in some cases and not as well in others. So in conclusion, we're making substantial progress on single insect assemblies from low inputs of DNA. Recent hi-fi approaches yield near chromosome contig assemblies from nanogram amounts of DNA. We couple that with Hi-C for validation. Really, Hi-C at this point is doing less, it's less of an assembler and more of a validator of the assembly quality. So you're not having to take thousands of pieces and push them together with the Hi-C scaffolding. You're just taking several pieces and saying, you're adjacent to you, and I support this very, very long contig. We're working to release these assemblies in the near future as submissions get complete in NCBI, or going into NCBI in the I5K at NAL website below. So with that, I just want to thank the whole group. This only represents a fraction of them and are welcome to take questions. Thanks.
Thank you, Scott. That was fascinating work, and this will have hopefully far-ranging impact on ag genomics in the future. In the interest of time, please keep posting your questions to the Q&A box. I see questions coming in. We will deal with them at the end, and handing it over to Anna to introduce the next speaker.
Yes, thank you. Next, we're going to have Alice speaking about aphid parasitoids and some new assemblies that she has.
Great, let me just start sharing. Can you guys see my screen?
Yes, we can.
Great, thanks for letting me have a chance to talk about this. I have posted here the link to my webpage where I'll put these slides afterwards. So you're all welcome to take screenshots or whatever if you would like to. So today I'm going to talk about 2 genomes that we have recently assembled and published in the last couple of months. Everything I'm going to talk about is the work of a very large group, most of whom are listed here. So I just want to start off by saying that when I say we are a big group of people that have done a lot of different things. Aphid parasitoids are a special group of wasps who attack aphids, ultimately killing their hosts. And this is how a parasitoid differs from a parasite. And aphid parasitoids are actually very interesting in that they lay their eggs directly inside the aphid. And as the larvae develops, the aphid continues to live and walk around while this larvae is wiggling inside of it. Eventually, the larvae kills the host, leaving behind this aphid exoskeleton that we call a mummy. And the wasp hatches from this. It's a very cool life cycle as far as I'm concerned. There's very strong reciprocal selection on both sides of this life cycle because the aphid, of course, needs to go on to reproduce and to infect or take over your tomatoes or whatever, while the parasitoid needs to defeat the aphid so that it can go on with its life cycle. And I'm not going to talk about this today, but there's a very nice body of literature about aphids and their endosymbionts. And it is these bacterial endosymbionts that are facultatively housed in aphids that give them a lot of their protection. And so this is just one factor to keep in mind. But I am rooting for the parasitoids in this situation. And because I'm in an agricultural session here, I want to first point out a couple of things about them, being namely that they can be used for biocontrol. And you can go online right now and you can buy a vial of mummies and have them delivered to your house and put them in your garden or more often in a greenhouse. And so these have this very important role in agricultural settings. However, because I'm an evolutionary biologist, I'm much more interested in the counter adaptations that might make this difficult. And as you remember, you have this endosymbionts in the aphids that may make it difficult for the parasitoids to control them long term. So we have spent quite a lot of time looking at coevolutionary dynamics in this system. And I'm just going to give you two different slides to try and convince you that there is coevolution going on in this system between the parasitoid wasps and their hosts. So this is a graph showing the success of different parasitoids on different aphid hosts. So all you need to see here is that the line shows that some wasps are better at parasitizing some aphids, and that there is no one best host, parasitoid, right? To look at this a different way, here are three groups of bars from three different parasitoids. And again, you can see that there is no one best parasitoid that can infect aphids. So this is one of the pieces of evidence that supports the idea that these wasps are co-evolving in relation to their aphid hosts. So I'm going to present today the genomes of two different parasitoid wasps, the Aphidius urvi and Lysophlebus fabarum that I've shown here. They both attack aphids, as I've said, although they attack slightly different tribes of hosts. And just to give you two pieces of their life history that makes them quite interesting, Lysophlebus has this ability to reproduce both sexually or asexually, and we find wild populations of both, whereas Aphidius urvi only has sexual populations. In addition, Lysophlebus can attack aphids that are defended by ants in the wild, and they do this by mimicking the cuticular hydrocarbons of their aphid hosts, meaning that they have potentially adapted in their genome for this. So I want to talk a little about our assemblies. Compared to Scott's talk before this, we do not have quite the same amount of data. This was started a couple of years ago. And we have two different approaches because of the data in these two different assemblies. So for Aphidius urvi, the assembly was done in what would probably be a more standard pipeline a couple of years ago, where we did an initial assembly using Illumina reads with a variety of insert sizes, and then these were scaffolded with PacBio reads. For Lysophlebus, we had a couple of issues with the data, and I like to mention this because this is one of these cases of making the best situation out of data that you have. So we had just one insert size for our mate pairs and paired and Illumina data. But there were some issues with the distances between our mate pairs where we didn't really have faith in them to use them for scaffolding. So instead, we took the mate pair data and filtered it heavily for read orientation and quality. And we did quality filtering of the Illumina data. And then we used both of these to error correct the PacBio reads with proofread. One note is that proofread can take a very long time. We then used these long error-corrected Illumina reads to to assemble with Kanu and polished this with the Illumina data. In addition, for Lysophlebus, we took advantage of its haplodiploidy by taking a whole bunch of haploid sons from a single mother and using these to create some phased SNPs that we could use to put a lot of the scaffolds into linkage maps. So our assemblies are pretty good as far as length with what we would expect. There are about 5,700 scaffolds in Aphidius and 1,600 in Lysophlebus. And I mention this because this is probably a consequence of our different assembly methods, and this is important. But if you look at the number of scaffolds that are longer than 3,000 bases, they're similar in the number of pieces that they're in. So we think that overall the two assemblies are pretty comparable, but I think there's a bit more duplication in the long read assembly of Lysophlebus. We then used these assemblies to run through the predictive gene annotation pipeline of Maker, and this gave us 20,000 genes for Aphidius and 15,000 for Lysophlebus. And we had pretty good support of our CDS with transcriptomics. The difference between the two species is more based on having more transcriptomic data for one than the other. And now comes the annotation part by hand. So we have these predicted genes, but of course these machine annotations can make errors. And so I think it's really important, if you are interested in some gene families, to have looked at this by manually annotating some genes. And so we assembled a community of people with an online Apollo system where we could let people curate their genes of interest and do this. In the end, we ended up working across about 7 countries and many more labs than that. So many work hours later, we ended up with about 700 genes that have been manually annotated for the 2 genomes. And these now mean that we can, oh, sorry, things are popping up. These now mean that we can have a lot more confidence in the gene counts of the families that we have looked at manually. Just to highlight a couple, the desaturase genes are involved in creating the cuticular hydrocarbon profile of the wasps, and the loss in Lysophlebus could be associated with this aphid mimicry that they use to avoid being detected by ants. Both genomes were also missing DNA methyltransferase 1, and this coincides nicely with data looking at DNA methylation in both species that it is found that is almost completely absent. So this is really quite cool. But I put in my title that I was going to talk about GC content. So I really would like to try and present some puzzling things that we found about GC content that I think are really interesting. So the GC content of the entire genome was 25 and 23% for the 2 species. And this is actually quite low for an insect. It's a little bit higher in the predicted genes, and this is what you would expect in most taxa, but it's really low in the 3rd position of the predicted genes, all the way down to 10.7% in Lysophlebus. And this is really, really low. Just to give you a sample of this, these bars, I don't expect you to be able to read the taxa, but these are from the predicted genes of 42 hymenopterans that I downloaded from RefSeq, showing the span of the GC content of their genes. And you can see how low our 2 genomes are over on the very right.
3 more minutes.
Okay. And when we look at the codon usage in the 2 genomes, they universally use the codons with the lowest GC content. And this is really very striking. And this was brought up by Clement Kent 2 days ago, showing that there might, perhaps there's a cost to synonymous changes that would change which codon you use. So there are a few reasons for this. Small genomes can have lower GC content. This lack of methylation could no longer control the GC content or could no longer constrain it. Perhaps there's a low effective population size or some mutational bias, but none of these are unique to our taxa. So I just looked at a couple of things to try and figure out what could be going on. So this is just a PCA showing the codon usage that I showed you those bars 2 graphs back of our wasps along with some of their host aphids or their endosymbionts. And this is not similar across taxa. So there's not just some shared biological reason for sharing building blocks in the system, or at least not that I can find at a first look. Next, I looked at expressed genes. So the most highly expressed genes should be under the strongest selectional constraints. And I wanted to see if these had lower GC content. But in both species, if you look at their overall highest expressed genes, they have higher GC content. So this doesn't support that there's selection for GC content to be lower. Lastly, I looked at the life history specific GC content in transcriptomic libraries I had for Lysophlebus fabarum, comparing larvae in adults. And here I find that the GC content of the adult biased expressed genes is lower, whereas nitrogen content is not. So I don't know why adult biased genes would have lower GC content, but they do. And perhaps this has something to do with energy constraints. It's cheaper to make A's and T's, and adults need to fly around and mate and chase aphids and inject eggs into them, whereas the larvae are inside the aphids, eating the aphids and eating their endosymbionts and doing very well. Maybe there's some other explanation associated with bias. Gene conversion, which would explain our third position, or maybe there's something else that we haven't considered yet. So in conclusion, I think aphid parasitoids offer a really interesting agricultural application system where you can also study coevolution. Our curation of gene families has really laid the way for a lot of future work looking into this. And I'm very interested in the evolutionary impacts of these low GC content genomes. Both genomes are available on the BIPAA webpage to download and work with, and so we hope this will be available to a lot of people. And lastly, I just want to thank you all for giving me the chance to talk. There are some logos from all of our funding agencies, and I will stick around after the talks today for questions. Thank you.
Excellent. Thank you, Alice. And in the interest of time, let's just move on to the next speaker. Please keep on posting your questions to the Q&A box.
Wonderful. Yes, next we'll have Arun talking about evolutionary histories and population genomics of 2 species of lady beetles.
All right, can everybody see my screen? You good?
Yes.
Okay, perfect. Should I get started? So are we on time? Okay. Perfect. So thank you, everybody, and thanks so much for all the organizers for doing this. I hope everybody's staying safe. These are indeed some crazy times, but hopefully everybody's cranking through some fun science, as I have been. So I'm going to be presenting some of my recent work that we've been doing on contrasting evolutionary dynamics and histories using population genomics to understand the effects of augmentative biocontrol on 2 species of lady beetles. And so I just wanted to give a little, since it's an agricultural genomics session, just a little intro into augmentative biocontrol. It's basically a repeated release of a biocontrol organism into novel environments just to augment imported populations. They've been already imported, but you're constantly sort of, augmenting these populations. And there's lots of examples of these. Here are a handful of them. Dr. Dennis just mentioned one, which was, some of these, aphid parasitoids that are very commonly used against aphids, or you got greenhouse, whiteflies that are destroying, and so you have some of these wasps that are used against that, or you have mealybug destroyers, or Hippodamia convergens, which is the convergent lady beetle, which is endemic to North America, which is very commonly used against aphids, whiteflies, and scales. I'm going to specifically focus on 2 species, and so the first species that I'm going to tell you about today is Hippodamia convergens, which like I mentioned, is native to the Sierras. It's got this very characteristic convergent behavior, which leads into its name, where they're collected en masse during overwintering across, and they're reintroduced or augmented populations across North and South Americas. And here's a little tidbit. These were, I don't know if folks followed, about a year ago, there was this massive congregation of Hippodamia convergens, which was actually even captured in Southern California, right here where I'm at, which basically leads to the fact that they do indeed have long distance migrations during spring and summer. The second species I'm going to tell you about is Harmonia axyridis, which is the harlequin or multicolored Asian lady beetle. It's native to Eastern Asia, and since early 1950s or 1900s, they've been introduced or augmented across the world. And they're known to also harbor parasitic microspores, which are known to be lethal to other congenic species, and therefore they've become invasive throughout the world. So the big picture here was, can we use population genomics to decipher if indeed augmentative biocontrol using these two species is being successful or not. And how can we use genomics and a combination of fitness assays to inform how effective augmentation actually is. So that's been a long-term goal of mine and my lab and collaborators. And so we summarized a lot of these into this recent paper that was just published yesterday, where we've reviewed a lot of methods or methodological considerations in population genomics and how we can use that to inform biocontrol. So here's what we did. So we took, for convergence, we had 83 individuals that we sampled across 16 sampling sites in North and South America. And we performed dd-RAD-seq, so double digest RAD sequencing on a HiSeq at Florida State. And then we processed that data using iPyRAD. And then for Axoridis, on the other hand, we had 159 individuals. We sampled from 27 different sites across Asia, Europe, and North America. And we performed 2B-RAD-seq, which is a single digest method on an Illumina NovaSeq, and we processed that using the STAX pipeline. So then, so these were some of the sampling sites, just to show you where we got our samples predominantly from. This is convergence here, and so we had a bunch of samples from the western part of the United States, a bunch of them from the Midwest and the eastern part of the United States, and then we also had one population in Chile that is representative in this case of South America. Axoridis, on the other hand, like I mentioned, we had Axoridis samples from across 3 continents. And so we had a bunch of samples from Europe and the US as well as from China. So here are some results from sequencing statistics. Here for convergence, we ended up with about 215 million reads, about 2 million paired reads per individual. We ended up identifying, after filtering, about 1,125 SNPs after applying several filters, quality filters, including the minor allele frequency filter of about 0.05. With Harmonia, we ended up about 1,230 million reads, about 3.8 million paired reads per individual. We identified about 1070 biallelic SNPs, again, post filtering. And so I'm going to share some of the results that we've been finding across these species. The first one that we identified is that in convergence, we identified unique clustering across a Western population and an Eastern population across its range. But if we split that into 3 subpopulations, then we identify the Western and then the Eastern, and then the Chile population actually stick out as 3 separate identities. So then we went ahead and we estimated the evolutionary history of these. And so what we see is clear support for this sort of a pattern where the ancestral population split off into an Eastern population, and then the Western and Chile populations were then therefore thereby established with little to no gene flow between them, which we also see when we estimated current contemporary gene flow across these populations. So even though we are doing a lot of augmentation, there doesn't seem to be any gene flow across these populations. So that's kind of the take home here. And so we went ahead and we did a bunch of fitness assays to see if these were actually being different. Are the Eastern and Western and Chile populations different in how they depredate aphids? And we also performed fitness assays using F1 hybrids. And so here's what we found. As it turns out, there's no significant difference in pupil weight or net weight gain ratio in Eastern, Western, or F1 hybrids across its range. So there's also no significant differences, even though they are genetically distinct. So that's pretty interesting. We just published this, or actually it's accepted right now at Biocontrol. It's out on bioRxiv. So really, is augmentation working? Maybe not. Are hybrids more effective? Maybe not. Okay, so that's with convergence. The second species, we went ahead and we analyzed the same kind of, did the same kind of analyses with Axoridis or Harmonia. And here we noticed that the clear split actually comes across Eastern, so this is Eastern Asia populations, and then the United States populations. And then if we split it even further into 3 separate populations, what we notice is that the European population actually sticks out as its own as well. So then we estimated
2 more minutes, please.
Rates, got it. Between these populations, and as it turns out, there seems to be no migration between the Asian populations and the Americas, but on the other hand, the European population is what seems to be establishing or having consistent contemporary gene flow between the American and Asian populations. So then what we went ahead and did was we looked at mitochondrial haplotype distributions across these populations, and we found that there's actually 2 unique haplotypes that we identified across the East Asian populations. And as it turns out, only one of those haplotypes is actually invading throughout Europe as well as the United States. So we went ahead and we did some fitness assays looking at fecundity per female, larval duration, female pupil weight, and male pupil weight. And as it turns out, the haplotype that is most frequently found in the United States and in Europe happens to have higher fitness across all of these characteristics, which is pretty interesting. And so therefore, we're continuing to study these in the context of genomic differentiation and trying to understand what exactly are the signatures of selection as a result of augmentation of these populations across the world. So are the two identified haplotypes different in this case? Yes, they are, right? So haplotype 1 happens to look to be more fit and found exclusively across its non-native range. Is augmentation using the species really working? Yes, considerable degrees of gene flow seems to be detected across the range in the world. So where do we kind of go from here? Oh, I just wanted to mention some challenges. Couple of things that I wanted to say about, so we did do all the population genomics processing using this pipeline for population genomics that we've developed. And you're welcome to go check it out. It's called PPP, stands for Population PopGen Pipeline Platform. Here's a link to that. There's a preprint out as well on that. All data will be made available on Dryad once we get everything published. Also, please do go check out our review paper, which covers a lot of how do we use pop genomics to inform biocontrol, which just was published yesterday. So where do we go from here? We have a lot of sequenced genomes. We have a Hippodamia convergence genome. We have a Coccinella septum punctata, which is the C7 or 7-spotted lady beetle genome, as well as a few parasitoid genomes. And I'd love to collaborate with more folks to work on these. So please do hit me up or send us a message on Slack or in the chat window, and I'm happy to talk more about our findings. So with that, a lot of funding agencies to thank for, I'm funded by, my lab is funded by the USDA and NSF. My collaborators are also funded by the USDA, as well as folks that did all the wet lab and the dry lab work on all of these things. So I think I did very good on timing. Wow, I have a timer in front of me, so I'll stop there. Thank you.
Great, thanks, Arun, and thank you especially for mentioning challenges. We were encouraging speakers to mention it. Once again, questions at the end. Quickly moving on to the next talk.
Yes, up next we have Rosanna and Ravikiran talking about genome and world population diversity of a soybean aphid population. And we can see your...
Okay, good. Today we are presenting the genome and population analysis of the soybean aphid, which was recently published in Insect Biochemistry and Molecular Biology journal. So we have sequenced and assembled soybean aphid genome using Illumina and PacBio data, and then used the genes annotated to perform phylome analysis using publicly available aphid and insect proteomes. We used the genome assembly to identify SNPs in North American population, and we developed a SNP chip, and then used that SNP chip to genotype several populations across the world. And then using those genotypes, we performed population genetic analysis. Rosanna?
Relies on alternate host, a summer host, which is the soybean plant, and a winter host, which is Rhamnus, multiple species of Rhamnus. And Rhamnus is used both as a place to reproduce and lay eggs. Next slide. There's a close association between soybean aphid and the soybean plant. As you probably all know, the soybean plant has been grown in China for 4,000 to 5,000 years. It has spread to other Asian countries from about 2,500 years ago. Both the plant and the aphids are widely distributed from Eastern Russia to the Philippines, likely facilitated by the cultivation of the plant. However, recently, because of global movement of people and goods, the soybean aphid has also been introduced to Australia and North America. In North America, it arrived in July of 2000, likely before that, but this is when it was first observed in Wisconsin, Illinois, and Minnesota. And within 4 years, it spread to 22 US states and 3 Canadian provinces, and is now very well established in most of the soybean growing areas in North America.
Comparison of available aphid genomes, we found that the Hongmei is the best of all the assemblies of aphids that are available in terms of smaller number of scaffolds and N50 scaffold length and the longest scaffold size. Our assembly is the next best assembly. In terms of genome size, Apisum is the largest and the smallest is Agocipii. We know that Agocipii is associated with a wide variety of host plants. So to become a super pest, you don't need a larger genome. And this is a PCA plot we generated using 926 SNPs, which are a subset of SNPs with minimized ascertainment bias. Using genotypes from all the samples across all different time points. When we zoom in here, we see that the China overlaps with almost all the populations except Japan and Australia. And Australia is closest to Indonesia, which could be its likely source. And when we look at the FST table, these results concur with what we found in the PCA plot, from the PCA plot. And this is a neighbor joining tree, which shows overall relationships among these populations.
It's of value to compare the original invasive habitats of the soybean aphid. And when we think about that, we can see that in Asia, there's very varied topography, mountains, bodies of water, whereas in North American Midwest, there's basically a prairie flat landscape with no barriers. Also in its original habitat, there were and are large number of natural enemies, whereas in North America, fewer natural enemies. Also to consider is that in North America, once the soybean aphid arrived, it was met with a barrage of pesticides, biological control, some effort of biological control, and attempts to develop resistant plant varieties, which have not been widely deployed. But nonetheless, within even with low deployment of these varieties, there were, in a very short period of time, biotypes of aphids that were virulent on these varieties of plants were observed. Biotype 2 on RAG1, 3 on RAG2, 4 virulent on RAG1 and 1 and 2. I just want to emphasize that the genome that we have sequenced is to biotype 1, which is a susceptible variety that was collected and established in the lab shortly after its arrival in the US. Here we see an examination of US and Canadian population using the entire complement of our SNP chip, 2,380, and a comparison of individuals collected across the US and Canada for the time periods of 2001 to 2013. And when we look at this data, what we see is that time is a better predictor of genetic diversity than geography, and that there's been a shift and reduction in genetic diversity through time, possibly as a result of the soybean aphid meeting the challenges of pesticide-resistant soybean varieties and the landscape itself. Next slide.
2 more minutes.
Also of importance is to consider the distribution of rhamnus in the US. As you can see from this map, the highest concentration of rhamnus is in Minnesota and Wisconsin. And we have looked at soybean aphids collected from experimental plots of susceptible and resistant plant varieties that were in areas close to rhamnus. As you can see here, the blue, and areas further away in yellow. Next slide.
The FST values in the heat map, we see that the FST values are high for rag varieties collected in Minnesota, Iowa, and Wisconsin. So Minnesota, Wisconsin, and Iowa are closest to large overwintering rhamnus, which serves as a reservoir of locally adapted virulent soybean aphids, which can recolonize soybean the following year.
So if you take a closer look at those aphids that were collected from the blue localities and the yellow localities, namely close to rhamnus, far from rhamnus, what we see is that the localities close to rhamnus have high numbers of clonal aphids, whereas the ones further away from rhamnus have unique aphids. What we hypothesize is that, as Kiran just said, in the localities close to rhamnus, aphids that had developed virulence towards resistant varieties were able to easily overwinter in the rhamnus stand, and then their progeny could recolonize the soybeans once they were planted the following year. Whereas the ones in yellow localities were unable to develop clonal offsprings in those resistant varieties. Next slide. And also what we see from the aphids that were collected on the resistant plants in the areas close to rhamnus, what we see is that the genes that overlap the SNPs with high FST values in a comparison between susceptible and rag varieties, what we see is that the GO terms that we identified are critical components of iron homeostasis pathways. And these were observed in RAG1 and RAG1 and 2, which are the harshest environment that soybean aphid was exposed to. And we hypothesize that these pathways are enriched in the soybean aphid due to perturbations resulting from plant resistance mechanisms. If you have any questions, we'll be happy.
Let's keep the questions for the end because we're a few minutes over right now. And let's move on to the 5-minute talks because the transition time is very little now.
Yes,
but
now we'll have Saeed talking about small RNAs in the Asian citrus psyllid.
I can share my screen. Okay, can you see the slides?
Yes.
Okay, thank you so much for the introduction. Today I will share my research on the role of the small non-coding RNA in the interaction of the diaphragm citrus with its bacterial community. Diaphragm citrus is the vector of the bacterium called Candidatus liberibacter asiaticus, CLAS in short, which causes a very destructive disease called citrus greening on the citrus trees. Once a tree gets that bacterium, there is no cure. In addition to CLAS, the diaphragm citrus harbors 3 endosymbionts, called Wolbachia, Protella, and Carcinella. All of the endosymbions are localized in the specialized organ called bacterium. Protella is localized at the center of bacterium in a giant syncytial cell, while Carcinella is localized at the surface of the bacterium in the individual cells. My previous research showed that the CLAS infection can alter the abundance of the endosymbionts in the host. That proposed a kind of interaction between the host and the CLAS. I hypothesized that this interaction is regulated by a small non-coding RNA. For this purpose, my research program is centered on trying to figure out how all these players use a small non-coding RNA to interact with each other at the molecule level. To study small RNA, I made 5 CLAS infected colony and 5 CLAS uninfected or healthy colony. For each replicate, I collected the nymph and adult from same colony to minimize the effect of sampling on the final data. Then I extracted a small RNA, I made a library, and I used Illumina HiSeq to sequence my library. Once I got the data back, I did the bioinformatic analysis, which I will share the data with you in the coming slides. The first and important analysis to check the data is the PCA, which recognizes the source of variation in the samples. The PCA analysis for me showed that the first source of variation is the development, the difference between nymphs and the adults. And the second source of variation is the CLAS infection, the difference between healthy and infected samples. By making sure that my PCA looks fine, I started to map all the small RNA reads individually to host genome and the bacterial genome. While this study produced many interesting results, for the sake of time, I'm gonna just share part of it. Strikingly, for the adult sample, about 50%, and for the nymph samples, about 20% of all small RNA reads map to the Protella genome. Its genome size is just 0.5 megabase, almost 1,000 times smaller than the host genome. And that pattern is specific to Protella, as I haven't seen it in the Wolbachia, other endosymbionts of the DC3. But more importantly, that's where all these distribute on the Protella genome. I found that all small RNA are not evenly distributed on the Protella genome. One of the hottest spots is located on the lysine tRNA gene. When I looked closer, about 600,000 reads with the length of 23 mapped on the lysine tRNA at the T arm, which proposed that these reads probably derived from the tRNA, which we call them tRNA-derived small RNA, or TRF in short. In previous study conducted in our lab, TRFs were discovered to be abundantly produced by the genome of the aphid bacterial symbiont called Bachnera aphilicola. Over 10% of the reads in that study mapped to the Bachnera-derived TRFs. The pattern of TRF expression changed depending on the aphid diet that you can see here. TRF also has been characterized from the bacteria that form symbiosis with the plant roots for the nitrogen fixation. The author showed that the bacterial cell can produce a TRF, which move out from bacterial cell to plant cells and match with the argonate protein to target and silence the host genes to initiate and promote the colonization of the bacteria in the root cells. That's an important subject of future research will be on understand how TRF can function in the symbiosis in the insect. I know that all of you are excited about the rest of the story, but because of the time limit, I'm gonna stop here, and I invite you all to watch for our upcoming publication. By that, I would thank all the HIC lab members and all of my collaborators, especially Surya Saha, for his help for the bioinformatic analysis, and thank you all for your attention.
Thank you, Saeed, for staying on time. And let's move on to the next speaker.
Okay, I'm gonna stop sharing. I think it's
next. We have Hongmei with Honeybee epigenomics, responding to viral infection. And we can see your slides coming up now.
Can you hear me?
Yes, we can hear you now as well.
How do I get the... Sorry, I'm trying to get the video. Okay. Yeah, so let me play the slides. So thank you for coming to my talk. This is a group of effort. So I want to just acknowledge everyone in the team with NC State and UNC Greensboro. And right now I am my own lab at Central State University. The research is focused on honeybee model system, because honeybees are a very good model system to study a variety of different topics, especially in animal behavior, ecology for pollination, as well as evolution and genetics, epigenetics. Within the honeybee colonies and population, there's a big concern on viral infections because the pathogenic viral infection, it can significantly affect the honeybee health for pollination. So one of the interesting topic is to study how the viral infections can interact and also affect the host honeybee epigenetics. So one of the main epigenetic mark is DNA methylation. So that's a cytosine nucleotide and methylated on the position 5, like the picture showed here. So we designed an experiment and kind of asked this interesting question: how is the viral infection can change the transcriptomic gene expression as well as methylomic changes in the honeybee host? So we use the honeybee pupa system because pupa is the very important developmental stage for honeybee life cycle, and it's a critical time for pathogenic and parasitic infections. So from the differentially expressed genes, we have detected a lot of genes in the later infection time. So we do have 5 hours as the infection, as the initial infection phase. And then we have 20-hour post-viral infection versus 48-hour post-viral infection. So by the way, the virus here we study is the Israeli acute paralysis virus. It's one of the main RNA virus occurring in the honeybee population. So very interestingly, we also detected a lot of the differentially methylated genes in figure C and D. So they're either hypermethylated or hypomethylated, but they are detected at the beginning, initial infection, and then significantly dropped the number at the 20-hour and the 48-hour post-infection. So we also look at the alternative splicing patterns, or we can call that differential transcript usage. There are a lot of genes detected under the infection. We detect shorter. So if you look at the graph on the big red dots, those are the significant ones we detected. They're shorter 3 UPR or longer 5 UTR, more on exon gaining, and also longer ORF, as well as less transcription starting site downstream. So we also look at the critical enzymes for DNA methylation, the DNA methyltransferase 1 and 3, and both of them have a significant drop at 20-hour as well as 48-hour post-infection. So we're interested in the role of DNA methylation in regulating immune genes. So we look at all the immune genes that are differentially methylated through all the time points, as well as their methylation. So it's quite interesting. We see 38 genes that are highly significantly differentially methylated at the 5-hour, which is initial infection time. But then at the 48-hour time, they have a significant gene expression changes. So this is the kind of first evidence now to show potentially the DNA methylation can affect the gene expression in honeybees, which is so in summary, we see the dynamics of the honeybee pupil immune response in a temporal manner after the viral infection. And interestingly, we saw we detected a higher number of differentially methylated genes and at the beginning, and then more differentially expressed genes in the later after infection. So a lot of genes are involved in immune response, such as the MAPK or JAK-STAT immune signaling pathways. So with that, I just wanted to let everyone know we're hiring a postdoc right now. So hopefully you can check out the posting and join us. So these are the details for the material methods.
Thank you. And let's move on to Zach's talk.
Our last speaker is Zachary with the improved Colorado potato beetle genome.
And please keep the questions coming.
Yes. Is that sharing? Okay.
Wonderful.
Great. Thank you for that introduction. And we're almost there. Just 5 minutes left, everyone. So sit tight. Won't regret it. And also thanks to the organizers for giving me this opportunity. This has been a real success so far for everyone. So I'm a graduate student working with Sean Scoville up at Madison, Wisconsin, and I'm interested in evolutionary genetics and genomics, and a lot of that is sort of contingent on a quality genome. So we really were motivated to improve upon our 2018 assembly. And that came about after attending a talk here through the I5K webinar series that was carried out by Adam Filippi. He was introducing this trio sequencing, trio binning approach to breeding and sequencing that we felt was really amenable to our system. Colorado potato beetle is a diploid sexually reproducing. It's got a relatively short generation time unless it goes into diapause at about 28, 35 days. So we wanted to apply this using this approach. The trio refers to the 3 individuals that will be sequenced as opposed to just the 1. But the result is you get 2 high quality haploid assemblies. So the parental individuals are sequenced at moderate coverage using short read paired end Illumina data. And their offspring, which needs to be heterozygous, at least 1%, which is fairly easy to achieve through inbreeding, is sequenced at high coverage with long read. We use the PacBio Sequel II platform. Now, given the beetle's biology, we know more or less that the ancestral range is the plains region of the United States and Mexico. So we collected individuals that were found on a natal host plant in that area, Solanum rostratum, buffalo burr. We maintained them in lab for several generations to reduce allelic diversity. And then we did the same thing with individuals that were collected on potato in Maryland. And those were where we derived our parents from. Okay, just a bit about what actually was carried out. Installed Canoe version 1.8. I believe they have 2.0 already. On the university cluster, it can be run on a single workstation or it can be multi-threaded depending on your resources. So that's convenient. It still took a bit of time to run, but it really wasn't the end of the world. We weren't waiting for that. What else, is there to do? So here's a little animation on kind of what goes on in the black box of Canoe. It's going to identify which parent those long read sequences were inherited from. And then it's going to segregate them into 2 bins, as it were, and run an assembly on each of those bins independently, and then produce two haploid genomes. So that's what we got here from that whole procedure. You can see that the first thing, I'm just gonna go sequentially, the size increased about 30%, which was kind of a shock. We weren't expecting to see it go up. Estimates of the genome weight based on C-value actually suggest something in the 400s. So that was, you know, whatever, it happens. But the other metrics really improved quite significantly. The BOOSGO for the dam went up. It's the sire and the 2018 assembly are pretty consistent, but you know, the N50 and the number of contigs are really what we're, you know, advocating this technique with. So if we kind of put all those metrics in the context of the I5K ranking as of October, we'll see that the CPB kind of, you know, skyrockets up to the top, as it should. It's its time. And yeah, that's all well and good. The next thing that I wanted to do, just very kind of quickly, was to identify where those gene features from the original assembly were in one of these. So I used the female from the dam, the haploid, it had slightly larger N50, and mini-map too. So I mapped all those gene features to that new assembly just to see what was there, what wasn't. And you can see most of those features are there. Again, this is the well-annotated, highly curated gene set, and they're on 70% or so of the scaffolds. So the next step is to maybe use those anchor points to scaffold and link them even further. So I just want to thank everyone again. We've made it. This is the end of it all. My advisor, Sean, and Dave Hawthorne for tramping out with me in New Mexico and collecting, and my funding stream through VPGA. So thank you very much.
And thank you, Zach. So we are a few minutes over, but that's okay. We had cameos from a few cats, so that was important too. Let me start off with a few questions for the 5-minute talks, and then we will go on to the longer talks. So for Saeed, do you want to talk about what kind of challenges you had in your sample and library prep, especially since you were trying to enrich for endosymbiont small RNA, which would be in pretty low titers? Yes, we can hear. Oh, no, you got muted.
Okay, you muted me again, but now it's good. So yeah, that's a great question, because I spent almost 6 months to figure out that how I can do that. I tried so many different kits, including the Qiagen, Sigma, and Trizol or whatever, but eventually I came up with the Mirvana kit that it's worked great. And after extraction, I could see the small RNA, microRNA bands clearly, which was really helpful to make sure that, okay, now probably I do have this small RNA from the endosymbionts as well.
Okay, great, thank you. And there was a question from M.L. Allen. Are the ACP small RNAs available as TSAs?
Not yet, because we are publishing the paper, so they're gonna be available soon on the citruscreening.org if you guys don't know that website, and the NCBI.
Thank you. Moving on.
My
to
cat
Hongmei.
name is
Yep.
a squirrel, by the way. Yeah.
So good. All right, we have a question for Hongmei from Amanda Stalk. Do you have any idea if these epigenetic changes are heritable?
So yeah, that's one area we try to study now. Right now in the honeybees, we know there are parent of origin genes, which are potentially for imprinting, but as of DNA methylation, we don't know anything yet. So,
okay.
Yeah.
And a question from Dorrit What is the infection cycle of IAPV in honeybees, and how is the virus transmitted?
Oh, okay. So can I go back to the first question? I had to add it into that a little bit. So
Sure.
there are, so two of the enzymes, the DMT1 is the enzyme that actually, during the DNA replication, the DMT1 is in charge of repeating in the daughter strands to have all the DNA marks from the parents. So from that point of view, yes. So each DNA, when it's replicated, and they are replicated in the DNA methylation pattern as well. And then DMT3 is the other enzyme will, depending on the cell signaling and cellular activities, and the DMT3 will add on new DNA methylation marks. Yeah.
And I'll repeat the second question.
Yeah, the second question about the IAPV, right? So the infection of the IAPV, basically, these are the RNA positive single strand viruses. They enter the animal cells by endocytosis, and so in animal cells, and when they're inside the cell, the positive strand is released into the cytosol and then translated by the host ribosomes. So the virus will use the honeybee ribosomes to make a lot of viral replication proteins. That proteins and will recruit the positive RNA to form a complex. So it's called a viral replication complex. And with that complex, the small amount of negative strand RNA will be produced. And then that will be able to use as a template to make to synthesize more large number of positive RNA. So that's kind of the cycle of the viruses. So in terms of transmission within the honeybee colonies, they have horizontal transmission from bees to bees. All the worker bees touch each other. And also from trophallaxis feeding each other and the feces touching, contacting. But they also have the vertical transmission from the queen to the, transmit to the eggs and then to the individual bees, during development.
All right, okay, thank you. And Zach, can you tell us a little bit about how you think the original genome will be used, considering that now you have a better quality genome and the scaffolds without genic features? What are they, do you think?
Yeah, for those that don't know, I think that's very common when you run that canoe assembler. Yeah.
Yeah. So to answer the first question, so the reference that we produced in 2018 was from Long Island, and that's like the most sort of pestiferous population. It's highly resistant and really destructive on potato. So that's still like a novel resource with respect to the individuals from this cross from Maryland and Kansas. And then, yeah, for the sort of other scaffolds there, yeah, I don't know. I think Anna alluded to some knowledge about what they might be. I'm actually not too familiar because I ran blob tools just to see if they were microbial. I think there was one out of the 200 or so, and the other ones were really kind of just insignificant in size as well. So they were all like less than 5 kb. So stuff like that.
Yeah, I think often if you look at those, you'll see a lot of repeat structures and other things that get pulled out. Canoe tends to assemble everything, which it can be good, and it can be, but you're left with kind of making some of those decisions about whether what those small pieces are. If we can move on to Rosanna and Ravikiran, there's a question here. What were some of the limitations that were encountered due to the paucity of genomic resources for your non-model organisms, such as aphids?
Yeah, especially with gene annotation, we have to mainly based on the annotations from human and other model organisms. And same thing with functional annotations. Basically, that is the main limitation.
I think we also have another question here. Is there any evidence, genomic or ecological, that the invasion of the Rhamnus carthitica through the US is impacting the soybean aphid? Any thoughts on that?
Rosanna, you want to answer that?
Yeah, I think maybe it, can you hear me or?
Yes.
Oh,
No.
okay. Yeah, so definitely the presence of the overwintering host has facilitated the establishment of the soybean aphid. Had there not been an overwintering host, obviously that would have been very difficult. So yes, the presence of Rhamnus definitely has facilitated soybean aphid being established. And also, as you saw from our results, for the regions that are close to these concentrated stands of Rhamnus, it is easier for resistant or rather virulent soybean aphid to survive the overwintering stage and be able to recolonize soybeans in the spring. So yes, Rhamnus most certainly affects the dynamics of what the soybean aphid can do in North America.
Great. Arun, we have a question here for you. It was interesting that the augmentation wasn't working with convergence. Do you have any idea why it's not?
So that's a great question. And we've been kind of breaking our heads about it too, because we've been trying, there's no evidence of any hybrid incompatibilities between the Eastern and Western populations, or, our fitness assays only detected things like just no differential fitness across feeding behaviors. But there are other phenotypic differences, including diapause times, mating periods, as well as like photoreceptor periods and things like that. And so my guess is that it has to do something much more phenotypic. So there's probably some degree of local adaptation going on in, the Eastern versus Western populations. And then the augmentation of the Western, you take the Western and you stick them somewhere else. We know that there's very long distance migrations that happen in the species. So my guess is they're just flying away and not doing what they're supposed to be doing. So there's also some evidence to that effect, actually. There was some research, I actually saw this at an entomology meeting a few years ago, where there's some research about a chemical called convergine, which is exclusively produced only by Hippodaemia convergens, which actually acts as a sort of like natal homing kind of situation, where they leave this convergine and these mass migrations actually follow this path of this convergine, which is pretty interesting to me. And so our goal, actually, this is also another question that was on there: why exactly are you sequencing the genome? Our goal is to sort of also to understand sort of molecular mechanisms behind migratory patterns in these species. And so, we would, ideally love to find the genetic basis of a lot of these long scale migrations as well. So those are some of my guesses. So, local adaptations, long scale migrations, and perhaps some degree of failure for hybridization. So we're looking for signatures of natural selection across the genome, as well as in our RADseq data right now.
Great. Alice, have you considered how ant attendance and its geographical variation could affect coevolution and produce a geographic mosaic, and the effects of this on genomic signatures?
I have not yet, but I would like to.
That's a great answer. All right, and we've got several more questions here. I know we're going to have to handle a lot of these online given the time. Scott, there was a question here about, does amplification of low input samples with that ultra low method, does that introduce any errors that you know of?
So I don't think the actual amplification process will introduce errors as much as difficult regions to amplify are going to impact the quality of the assembly. So if you're going through extremely GC or AT rich regions, you know, you're having to have a polymerase that can get through that. So these are high fidelity proofreading enzymes that should be doing everything properly, but there's probably a lot of amplifications that fail because it's just going through these hard regions. And so the way the process works is you actually set up parallel reactions that are geared towards high AT, high GC, and sort of normal DNA regions, and then you pool all those together to try to get the best representation of the genome as possible.
All right, I think what would be best now, given the time, is to hand this over to Gloria and then Sue, who have some closing remarks for us.
Well, if I could just reiterate the thanks to the rest of the organizing committee. We managed to make it work, I think, just about as we had planned, making it up as we went along. I also would like to thank everyone, all the participants who actually read the user guides and therefore didn't have any problems connecting or understanding how to participate. Sue already mentioned the future of the AGS Slack workspace. So we really hope that it evolves into something that really serves the whole community. And so you're welcome to go ahead and invite colleagues who were not necessarily registered for the symposium to join the Slack workspace, and we'll accept those invitations as they come through. And the last little reminder that there is a plan for an informal discussion chat happening once we've finished this session in about 15 or 20 minutes for anyone who wants to keep asking questions or just wind down a little bit after the hecticness of this week.
The Slack will remain open. We will be asking you more questions in the post-conference survey about how you would want to use Slack, if we can make it better for you. But please, you can form small groups, I think up to 10 people, where you can keep on communicating on research topics, pretty much anything. And of course, you can message anybody directly. If you don't use Slack, please make an effort to check once in a while, because somebody you wanted to talk to may have posted a message and you just don't get a notification because you don't look at Slack.
So I'm really excited. I think this was a wonderful format, a wonderful venue, and I was really excited to hear so many great talks. I think that we did a really awesome job of getting together a really truly global group, and we had much broader participation because we were able to do this virtually. And I'm really excited to see this, and I hope that we can do some kind of hybrid method in the future if we ever do get back to a normal meeting format. I think it's really important to acknowledge our organizing committee again. And of course, we have to start with Rob and Sharia. They have just done a phenomenal job of putting together the Slack and the Zoom and having all of this go through very, very, yes, I agree. Everybody, I think, and all the participants can agree with me there. I would also like to acknowledge the rest of the organizing committee, including Christine Panfilio, Anna Childers, Mike Frender, Fei Lee, Gloria Geraldo Caldera, Mary Ann McDowell, and Josh Benoit. Did I leave anybody else? I hope not. I think I got everybody. Good. They've been doing an awful lot of meeting. They've been meeting at 7:30 in the morning my time, which is not a good time for me because I'm just getting out of the gym. So I missed a lot of them, but I was still able to get into the last little bit and help run a session. So I think that's exciting. I think all of us are excited, and I would like to continue a theme about the workshops. If you really like the way this is working and you would like to see more workshops, like maybe you would like to see another Apollo workshop in a month. Sorry, Gloria, or 2 months. So if that's true, we can organize them through this, and we can have people coming on and learning to do it. We love having peer teaching. And so I have a lot of people that use Apollo in my own lab. And so we love having the undergraduates teach the next upcoming undergraduates. And they have a lot of novel ways of doing this. And so I think there's a lot of information we can convey through this Slack and through continued either webinars or just Zoom meetings that are not quite as big as the entire community. I, for example, would like to see a workshop in the next few months on a virtual kind of workshop on genome project management. I think a lot of us have a lot of questions about how do you go from sample collection to NCBI submission or some other database submission. I think we all really want to get there, and we all want to have the Earth Biogenome Project style format for our genomes and the quality control that brings. But there's new tools coming out all the time, and it's often fun to have the authors of the tools come and present them to us, as well as the people who are using them. And getting those two together has worked a lot. One of my students has become a beta tester of the blob tools just because he's trying to use them. And so I'd like to see things like that going together here. With that, I would like to say that I'm sorry we didn't have swag like we usually have at the in-person meetings, but hopefully maybe we can have some kind of download decal that we can all print out or something, put on our coffee cups for next time. There's one other type of channel we might have is to have families, orders, your group, so that you can get together and work with the people that are working on the same species you're working on or the same biological questions you're working on. So with that, I would like to close by asking you to think carefully about the questions that you might, and the answers you might give to the survey. And please, please, please fill out the survey that comes. It won't bother us to get 1,000. So we're really happy and we really want your input. So thank you all for coming to this virtual meeting. It was nice to meet some cats and dogs as well as kids and other people. And it's nice to see everybody. And I hope to see you again soon. Thank you very much.